So it's a very broad definition. So as you probably know, the people in the department, the rest of the country, do very vastly different things. So I guess I'll answer my piece of it, which would be computational material science. So I think the way it's most useful to think about breaking down is into kind of hard material science and soft material science. And so the hard stuff is kind of ceramics, metals, and similar to those guys, and the things that we're doing for biomaterials, so sort of proteins and peptides. And I guess the way that fits into the larger kind of umbrella of material science is interesting questions about looking at atomic and molecular length scales you can't really see experimentally. So I guess for a large part, computational material science can be useful at looking at very small things that can be probed experimentally and using that to help design and inform perhaps future experiments or future studies. Sure. So I mean, a lot of biomaterials could be thought of as kind of healthcare industry, things like that, looking at cellular things and growth media or growth structures for cells. I think what we have is a more molecular picture, and so we're looking at actually the biomolecules. So looking at single proteins or protein aggregation or single DNA strands and the way those interact with components of the body or the cellular matrix. So perhaps one of the most kind of low resolution, I suppose, things you can look at is a single molecule, a single biomolecule. And so we're interested in the ways that proteins, both their dynamic and the equilibrium properties, and the way that can be used for, say, therapeutics or for antibody design or for helping to understand perhaps biocatalysis. And so those very kind of atomic resolution things are sort of the biomaterials angle that we're interested in. I think the potential to help people. So biomaterials is very, I think, health driven, and so the ways to develop novel therapeutics or perhaps wound healing, vaccines is one of the big things we're interested in this group. So using kind of bioinformatic tools to figure out ways to design new vaccines. So one of the projects that we're currently working on is to use HIV databases in order to figure out the best targets for next generation HIV vaccines. And so that's very exciting to me, the potential to help people with that. In addition to, it's also kind of fascinating scientifically. So if you can blend both of those things together, I think you're going to have a great time doing biomaterials research. So I will say that there are three great things about this department, very generally. So number one is that the faculty in the department are really great. So everybody's very friendly, everybody talks to one another. And so aside from being a nice collegial place to work, you also have the opportunity to establish collaborations you may not have thought about before. So for instance, I was just talking to JJ in the corridor one day and Bill Wilson over at the MRL, and just chatting about things that seem to overlap, and some exciting things might come out of that. So it's a lot of fun to work with your colleagues when you have a nice collegial atmosphere. The second part of the students are really great, and so they're top-notch students, and they're also very friendly, and really want to get involved, like you guys, in the student societies and chapters. And so that's a really great thing to have in the department, is top-notch students are able to work with you and do great research. And then the third thing is the facilities. And so I'm a computational guy, being at the place where the National Supercomputing Center is a real great help for my own research. I mean, both the hardware and the folks around know how to run the hardware and the software is really wonderful. Sure, so let me give you kind of a super high-level overview, and I guess it breaks down into three parts. The first part I touched on already, which is vaccine design. And so what we're doing is using these models from statistical physics and kind of magnetism in order to fit these landscapes to describe the fitness of a virus as a function of its sequence. So basically we're trying to figure out vulnerabilities within the virus. So where do you hit the virus with some vaccine-induced immune pressure to make it as unfit as possible and make it able to be controlled within your body. And so we're interacting with some experimentalists and some clinicians to test our predictions, which seems to be going pretty well. And then the next step is to use these to kind of inform vaccine design. So that's a really exciting area that's really sort of bioinformatics and maybe biomaterials. The second component we have is self-assembly. And so we're using these very smart data mining and machine learning techniques to try and figure out how particles assemble together into interesting and useful structures, such as 3D photonic crystals, or perhaps patchy colloids, which can assemble into drug delivery vessels, or even things like viral capsids, how the constituent proteins come together. And using these data mining techniques, we can first of all try and understand better how these processes are happening, and second of all, use that understanding to help design better building blocks to assemble into things that we want, or inhibitors to prevent things from assembling that we don't want, such as viral capsids or amyloid plaques and things like Huntington's disease. And then the third component is more kind of computationally based, less applied, which is on accelerating molecular simulations. So again, using some nonlinear machine learning techniques to try and learn how molecular simulations are performing, so the particular motions that the protein is trying to execute on very short timescales, and then to accelerate along those motions to try and speed up the molecular simulation. So why is that useful? Well, you have a very big biomolecule, say like a surface receptor on HIV. You just don't have the computational horsepower in the world to kind of figure out all the different states it can interchange between. And so to accelerate the simulations using these approaches is very useful in figuring out its native state and figuring out the dynamical motions. And then again, you can use things like the structural information to design antibodies or drugs to try and interfere with that. So that's another area we're pretty excited about. So when I started grad school, which would have been 2005, I was pretty sure I wanted to go into academia, but wanted to also leave the door open to do industry. So I wasn't totally dead set on it. And so doing my graduate degree was chemical engineering, and so that can go either way. So definitely I had both tracks open to me, and a lot of my contemporaries and my colleagues decided to go the industrial route. But throughout my graduate school career, I felt that research was something that really appealed to me and really excited me. And I guess you feel when you're putting in the long hours in the lab that there's going to be some reward at the end of it. Fundamental scientific discoveries and helping develop new technologies, especially in things like vaccine design or protein folding understanding, or very applied things with kind of exciting implications. And so that really stimulated me, and the idea of having a self-directed career where you can work in a group doing self-directed research in areas that interest you seemed like a very exciting career path to me. Certainly the downside is it's not a 9-to-5 job. It does tend to consume your life somewhat. But as long as you're excited in what you're doing, I can't remember where the quote is from, but if you do what you love, you'll never work a day in your life. And so that's kind of the appeal of academia to me. So I guess it's like asking questions about top 10 songs or something. It's always the most recent one, right? And so I guess if I was being subjective, the most recent paper we're writing just now is on this vaccine design. And so using kind of these physics tools to help design new HIV vaccines. And so we're currently writing that one, and it's being submitted. And so we're super excited about that one. But I guess if I was to try and step back and be a little more objective, we had a really nice paper that was an experimental and computational collaboration working on this short antimicrobial peptide. And this peptide is very cool. You can boil it, you can expose it to like 6 molar acid, and it retains its activity and its structure. And so it's got lots of potential to be like a food preservative or a new antimicrobial antibiotic. And so working in a team with experimentalists and computationalists was really exciting to me, operating on 2 parallel tracks and finding results that reinforce one another. It was kind of stimulating for me to see the power of having experiments and computation working together. And so we had some really neat findings, and we figured out structure and different ways the structure could assemble, and inverter role for the maturation enzymes that help it work out its final structure. And so that was a lot of fun. I enjoyed working on that project a lot.